So good morning, students. So I welcome you again all in the lecture series of B Pharm 6th semester in the biopharmaceutics and the pharmacokinetics. So myself, Dr. Dilpreet Singh, working as an associate professor in the ISF College of Pharmacy, Moga. So in continuation with the topic absorption and the basic concepts of biopharmaceutics, so we shifted towards its advanced version, that is factor affecting the drug absorption, and we started with the part one, that is biopharmaceutical factors. So just let me introduce about the basic concepts like the absorption, bioavailability, and the biopharmaceutics. So absorption, as I already discussed with you, that is basically a transfer of the drug from its site of administration into the systemic circulation that manifested by its site of action and is manifested by the site of administration. So in general, what we depicts. So when we take the drug orally at the site of administration, the concentration is very high. So after different absorption mechanism, the transfer of drug was initiated from its site of administration into the systemic circulation. So that is collectively called the absorption. And then we shifted towards the advanced definition, that is bioavailability. It is known as the rate and the extent of drug absorption. So rate, it depicts the time. Extent means amount. So rate, it depicts the T max, C max, and then we have the absorption rate constant, elimination rate constant, and extent, it depicts the area under the curve, clearance, volume of distribution. So it is characterized by the rate of transfer of the molecule from one compartment to another compartment, and what is the total amount get transferred. So biopharmaceutics is the collection of the factors that influences the bioavailability and the drug absorption. So it is the collective term which defines the bioavailability and the drug absorption in a pharmaceutical oral product. So biopharmaceutics is concerned with the relationship between the physicochemical properties of the drug in a particular dosage form and what we get observed therapeutic response after the oral administration in a biological field. So biopharmaceutics, basically what is the definition is the basically the effect of dosage form and the route of administration on the biological effect of a drug. So when it's collectively called, what is the dosage form, what is the strength, what is the effect of the dosage form on the body, and what is the route of administration on the biological half-life of a drug. So it is the collective study of factors that it influences the presence of the drug at a site of absorption and the transfer of the dissolved drug across the biological membrane into the systemic circulation. So biopharmaceutical methods involves the knowledge of drug release and a transport mechanism which favors the predict therapeutic effect from a product upon administration to a particular patient. So it also gives us the knowledge of the release profile of the drug. It gives knowledge about the transport mechanism across the biological membrane to obtain the response curve. So it gives also the knowledge about the site of administration and site of absorption. So biopharmaceutics is basically a vast field. So the study of the factors, including the simple to complex terms, they are wide resources and they are basically a wide, vast scope in a particular biopharmaceutician and pharmaceutics persons. This is that a collection of the events in the absorption of the drug from orally administered dosage form. So when we taken the drug orally, the granules or aggregate were formed during the disintegration, and during dissolution, fine particles was observed in the solution form. So drug will go into the absorptive site, but in the solution form, and our biological lipids and endophospholipids, it converts the ionic drug to non-ionic state. So the non-ionic drug or ionic state basically is in the reversible order. So basically, the ionic drug that get been transferred from one absorption site to another absorption site and is goes into the blood. The non-ionic drug is basically protein-bound drug, and the protein-bound drug is basically reversible in nature. As long as the ionic drug was absorbed, the non-ionic drug will slowly being detached from the protein molecule and it's slowly absorbed from the gastrointestinal lumen. So basically, when we characterize a solid dosage form, it converted into a solid drug particles, and drug in the solution form, it will goes into the absorption site. So basically, the conversion of a solid drug particles to solution state is a rate limiting step when we talk about the lipophilic drugs. So lipophilic drug is basically water insoluble in nature, and so though its water insolubility and the lipophilicity, it imparts the greater effect on the bioavailability. So when the drug is in the solution form, it distributed in the body in tissue, different tissue compartments, and in the regarding the distribution of the drug in a particular tissue compartments, permeation is the rate limiting step for the hydrophilic drugs. So for the hydrophilic drugs, permeation is quite rapid. So rapid permeation, it influences the first pass metabolism, and drug it rapidly detached out from the tissue compartment, and then it excreted out from the body rapidly. So it also create a erratic bioavailability problem. So when we talk about the dissolution and permeation, it should be equally uniform and equally be been higher when we talk about the drug administered in the oral dosage form. So the dissolution and permeation is one of the important step when we taken the drug orally for the testing the oral bioavailability assessments and other lipophilic parameters. So when we talk about the poor solubility, poor permeability profile, so some of the scientists it create a classification system that is called biopharmaceutical classification system, and it classify the drug according to its nature. Class 1 drug is exhibited high solubility and high permeability, and it is well absorbed, and a rate limiting step is basically gastric emptying. Example including deltiazem, hydrochlorothiazide, then we have the diazepam is there, then we have the imipramine, amitriptyline. Class 1 drug exhibit low solubility. It is basically water insoluble nature, and it exhibit high permeability. The absorption is quite variable. Means it is basically up and down movement. It is basically it show peak fluctuation value. That is called peak and valley fluctuation. So rate limiting step is basically dissolution, and example including nifedipine, then we have the hydrochlorothiazide, acetazolamide, docetaxel, and what we have, another example, it including the amipramine, uh, ACE inhibitors like the amipril, ramipril. So the third classification, it including the drugs which categorized as a highly soluble drug, but it show low permeability. The absorption, it again quite variable, and the rate limiting step is basically permeability. So example including the insulin. The fourth class of drug is basically the drugs which encountered about 70% of its concentration. About 70% of the new chemical entities from the drug discovery and the lead approaches exhibited low solubility and the low permeability profile. So the absorption is basically quite erratic and is poorly absorbed. So rate limiting step is, I think it's around both. It's basically dissolution rate limited also, it is permeability rate limited also. So, for example, it including the docetaxel, around 90% of the anti-cancer drugs, about 90% of the aminoglycosides, and about 90% of the painkillers in the same molecule, it exhibited poor solubility. So, in definition, the intrinsic solubility is basically the maximum amount of solute that dissolved in a per unit of solvent under standard conditions of the temperature, pH, and the pressure. The dissolution rate is basically the amount of solid substance that goes into the solution per unit time under the standard condition of the temperature, pH, and the pressure. So when we talk about the applications of biopharmaceutical studies, it influences the key aspect of the developmental process and has been becoming the integral to the design and the development of new pharmaceutical oral products. The new pharmaceutical oral product, it including the drug substances design, formulation design, toxicological exposure assessments, biopharmaceutical studies that act as a key player to drug future success. So regarding the other application, then we talk about the drug discovery process. It also about application of biopharmaceutics, including the compound selection, preclinical efficacy and safety testing, other parameters including the formulation development, clinical efficacy study, and post-approval changes. At every stage in the drug development, biopharmaceutical scientist it plays a major role in multiple disciplines, including the chemistry and the biology, drug safety assessment, pharmaceutical development, regulatory affairs, marketing, and the manufacturing of a formulation for the oral delivery. So when we talk about the biopharmaceutical factors, the key factors that play a major role in the absorption, including the drug solubility and the dissolution rate, particle size and the effective surface area, polymorphism and the morphization, pseudopolymorphism, including the formation of the hydrates and the solvates, salt form of the drug, lipophilicity of the drug, pKa of the drug, including the gastrointestinal pH, that collectively called as pH partition hypothesis, and the drug stability. Pharmaceutical factors including the disintegration time, dissolution time, manufacturing variables, pharmaceutical ingredients, excipients, adjuvants, nature and the type of dosage form, and the what are the storage condition of the formulation. Biological factors including the route of administration, membrane physiology, what is the nature of the cell membrane, what is the transport processes, what is the age of the patient, what is the gastric emptying and intestinal transit time, and what is the disease state of the patient. So if we want to talk about the gastric emptying, that is what we need to do is to study the fed and the fasted state. So basically, the drug-food interaction, which we want to know, what are the effect on the fluids on the absorption, and what are the other normal GI content which primarily influence the absorption. So pre-systemic metabolism is also been affected by the luminal enzyme, gut wall enzyme, bacterial enzyme, and the hepatic enzyme. When we talk about the drug solubility and the dissolution rate, so the rate limiting step in the absorption of the orally administered molecule, they are basically the rate of dissolution and the rate of permeation. So dissolution is basically the rate limiting step for lipophilic drugs. Example including the glycofulvine and spironolactone. Permeation is the rate limiting step for the hydrophilic and the highly aqueous molecule. For example, the neomycin, bleomycin, aminoglycoside antibody, and the chromomin sodium. The theories of drug dissolution. So basically, dissolution is a process of basically a solid particle. It convert into a solubilized state in by some mass transfer reaction in the on our biological system. Basically, that is collectively called as a mass transfer from the solid solute to a liquid solute. So several theory was established to explain the drug dissolution. Some of the important theories like the dissolution layer model, that is film theory, that is called a Danvert model, and then we call the interfacial barrier model. Diffusion layer model or film model, this is one of the simplest and commonest theory of the dissolution. Here, the dissolution of the solid particle, it commences in two steps. First step is the conversion of the solid state to solution form at a solid liquid interface, that is called stagnant film. And the diffusion of that stagnant film to the bulk of the solution, that is basically the rate limiting step in the drug dissolution. So the more breakage is the stagnant layer, more steeper is the drug particle, and more faster is the dissolution rate limited is. So basically the formation of the stagnant layer and the formation of the solid liquid interface during dissolution is one of the key factor for incorporating the dissolution into a solid dosage form. So the rate of dissolution is governed by the Navier-Stokes equation, whereas these particles of the solid surface is getting detached when it contact with the solution interface. So the equation that lies is that DAC by dC by dt is equivalent to K Cs minus Cb, where dC by dt is equivalent to dissolution rate of the drug, K is the dissolution rate constant, C is the concentration of drug in the stagnant layer, Cb is the concentration of drug in the bulk of the solution at a time t. So the previous equation that were based upon the fixed second law of diffusion. What is a fixed second law of diffusion taken place that the fix that basically it coined by the Nernst and Brunner, and it modified the Navier-Stokes equation to modified Navier-Stokes equation, that is basically dC by dt is equivalent to dAk W by O Cs minus Cv divided by Vh, where D is the dissolution coefficient of the drug, A is the surface area of dissolving solid. K W by O is basically water oil partition coefficient of the drug. V is the volume of the dissolution medium. H is the thickness of the stagnant layer, and Cs minus Cb is equal to concentration gradient for a diffusion of a drug at a solid water interface. So when we talk about the fixed second law of diffusion, there is the creation of the Singh condition. So Singh condition, it represents the what we call as the replacement of the fresh fluid during dissolution of a particular solid solution. So non-Singh condition, it represents the first order dissolution rate process, and the driving force for which the concentration gradient generated is dependent upon the time interval. It dependent upon the how much solid surface is created. It dependent upon the how a stagnant layer is solid is. So if we talk about the stagnant layer, the dissolution is said to be under non-sink condition. So when we talk about the sink condition, so the in vivo dissolution is always rapid than in the in vitro dissolution, because when we talk about the in vivo, the drug dissolve in the stagnant layer, and the dissolution is much faster as compared to in vitro. In vitro, it is absorbed in the systemic circulation. So as a result, the concentration gradient is equivalent to zero, and the dissolution is occurred at a maximum in the sink condition than the non-sink condition. So in the sink condition, the basically first order dissolution rate is created, and in the non-sink condition, the zero order dissolution sink sorry, the zero order dissolution is the created at a maximum. Second theory is basically Danvers theory. The Danvers theory is taken into account basically the eddies or the packets that are present in the agitated fluid, which undergo solid liquid interface interaction, and it absorbs the solute by diffusion mechanism, and it carry into the bulk of the solution. So these packets get continuously replaced by the new ones, and when it exposed to the solid solution, the formation of a thin film that is called stagnant film is there, and the stagnant film generally burst up and it convert into a small packets. These packets containing the drug, it leaving the interface, and the solute particle it rapidly dissolved and is available for the absorption. This is basically also called a surface renewal theory. The third theory is basically the interfacial barrier model, that is called double pair or limited solvation theory. So the diffusion layer model or Danvers model were based upon the two assumptions. The rate limiting step that controlled dissolution is the mass transport, and the solid solution equilibrium which is achieved at the solid liquid interface is always being directly proportional to the function of the solubility. So when we consider the dissolution of a crystal, each phase of a crystal will have a different interfacial barrier. So these interfacial barrier, it creates a noise on the film, and these film get disrupted and the dissolution is achievable at maximum. A- so this is all about the Danvers theory. So the question comes under this topic, that is basically enlist and the illustrate these steps that involved in the absorption of a drug from the orally administered dosage form. What is the rate limiting step in the dissolution and the permeation when we talk about the drug taken orally? Classify and enumerate the biopharmaceutical factor which influences the bioavailability of a drug from dosage form. Justify the statement, that is basically dissolution rate is better related to drug absorption. So when we justify the statement, dissolution rate is always been directly proportional to intrinsic solubility, and when intrinsic solubility is there, the rate limiting step, that is dissolution, is basically higher in the proportion, and there is no formation of stagnant film. So when we talk about the non-formation of stagnant film, the absorption is achievable at maximum because there is no discrete particle formation when dissolute the solid particle into the liquid solution. The drug is always uh, what we call is held in the solution form, and the solution form of drug is always get better absorption as compared to solid particles. So name the various theories that explain the drug dissolution, that including the interfacial barrier theory, Danvers theory, or what is called as the film theory. So using the noise-Whitney equation discusses the diffusion layer theory and the variable that influences the drug absorption. These are the MCQ. So when we discuss the MCQ, what is the critical rate limiting step of the drug absorption? The answer is C. That is the size of the porous particle. So which sentence will define the dissolution rate? So the correct answer is the B, that is amount of the solid substrate that goes into the solution under the constant time under standard temperature pressure conditions. The third question is the what is the factor affecting the drug absorption? It included the physicochemical properties of the drug, pharmaceutical factors, biological factors, or all of the above. So the answer is D, all of the above. So which one of these is not a theory of drug dissolution? Basically, the answer is the B, that is called Fick's law of diffusion. Fifth question is the passive diffusion process is best represented by which of the following? That is the answer is the C, that is the penetration or the surface renewable theory. So the next parameter that is called pharmaceutical factor. So in pharmaceutical factor, the disintegration time is one of the important factor to show a rapid absorption when disintegration time is required. So disintegration time tell about the rapid absorption. So it is directly proportional to the amount of the binder and the compression force. So in vitro disintegration test under standard Indian pharmacopoeia guidelines, it gives that the guarantee of the drug bioavailability when we talk about the disintegrated drug particles which do not dissolve until absorption is not possible. So it also tells about the what we tells about the quantification of the drug bioavailability and is also defined about the qualitative assessment of the absorption at in vivo rate. For example, the coated tablet they have the longer dissolution time. So fast dispersible tablet they have shorter dissolution time because of the super disintegrants and the binder concentration. So dissolution is a process in which a solid substance solubilizes in a given solvent, that is mass transfer from the solid surface to the liquid phase. So what are the manufacturing variables? So several manufacturing processes it influences the drug dissolution from the solid form. That is like the method of granulation and the compression force. So on the other hand, when we talk about the compression force, higher compression force is causes the deformation, crushing, or the fracture of the drug particles into the smaller ones. So it causes a larger increase in the effective surface area. So this surface area increasement results in an increase in the dissolution rate of tablet. So a combination of the curves A and B is also possible as shown in the curve C and D. So when we talk about the differential compression force, the rate of dissolution is quite steeper. So when we talk about the integral compression force, the drug dissolution is quite high. When we talk about the antipode or sympod compression, the dissolution is quite variable and it show the zigzag motion of the dissolution. So when it call about the non-compression, the dissolution rate is always been declining as a result of the improper binder or disintegration concentration. So what are the pharmaceutical ingredients? Also the ingredients in the tablet that impart a major effect in the dosage form, bioavailability, and the absorption. So what we call as the more the number of the excipient, more greater is the potential for absorption and the bioavailability problems. So commonly use हम करते हैं excipient which is used in the various dosage form are the vehicles. The vehicle there play a major role in the filler or and in the bulk up of the tablet. So it influences the rate of absorption. It influences the partitioning behavior from the oil phase to aqueous fluid. It influences the rapid absorption from the solid dosage form to liquid dosage form. Example including the propylene glycol, carboxymethyl cellulose, or the some starch molecules also. B is the diluents. So diluents it imparts the bulk up of the tablet. The diluents basically there are two types: hydrophilic diluents and hydrophobic diluents. Hydrophilic diluent it imparts the absorption, while hydrophobic diluent it retards the absorption. Also, there is a drug diluent interaction. It forming the insoluble complex and it retards the absorption. For example, tetracycline. So the third C basically the binders and the granulating agent. So the hydrophilic binder it imparts the hydrophilic properties to the granule surface and it gives the better dissolution properties. For example, starch, gelatin, PVP. So more amount of binder it increases the hardness of the tablet and it retards the absorption rate. When we talk about the disintegrants, disintegrant basically is hydrophilic in nature and decrease in the amount of disintegrant significantly lowers bioavailability. This is because that amount of disintegrant is sufficient to need to dissolve the molecule from solid surface to the liquid surface. So the if in general when we already tells that drug is in the solution form, it exhibit higher bioavailability as compared to the drug which is come into the solid surface. So decreasing the amount of disintegrants, it always restrict the lower bioavailability problem and it also gives the erratic bioavailability orally. E part is basically the lubricant. So basically, it is hydrophobic in nature, basically water insoluble, and therefore it inhibits the penetration of the water into the tablet, and thus it it creates a dissolution and the disintegration inside the body. Suspending agent is basically it stabilizes the solid drug particles and thus it affects the drug absorption. So the macromolecular gums it form the unabsorbable complexes with the drug that is basically called as sodium CMC and the viscosity imparters and basically the viscosity inducers that act as a mechanical barrier to the diffusion of the drug from its dosage form and it retards the GI transit of the drug. So basically, the different excipients like the viscosity improvers, macromolecular gel surface stabilizers, and the gelation properties of the gum, it influences the marked absorption behavior and the bioavailability when we talk about the drug taken orally. Surfactants. So surfactants basically it enhance the or retards the drug absorption depending upon the behavior of the surfactant, depending upon the head to tail conversion, or depending upon the HLB of the surfactant. So basically, the hydrophilic surfactant it improves the bioavailability, while hydrophobic surfactant it retards the bioavailability. For example, the glycosylvin and steroids. The glycosylvin steroid it containing the non-ionic type of the surfactant, so it increases the absorption when it forms the unabsorbable complexes with the drug above its critical missile concentration. Coating. So coating is one of the important parameter when we talk about the tablet taken orally. So different coating they are existed pharmaceutically. There are two types basically enteric coat, sugar coat, or non-enteric coat. Enteric coat exhibit greater bioavailability than sugar coat and then enteric coat. So the dissolution profile of certain coating materials it will change on time. For example, the shellac. Shellac basically it's having that prolonged stability and it dissolves more slowly in the intestine. So this can be prevented by the incorporating the little PVP in the coating solution, which disrupt the shellac coat inside the tablet, and then it promotes the absorption quite fast as compared to shellac alone. So it depends upon the which type of the polymer you have used for the entire coating or for sugar coating. Buffers they are basically sometimes used in the creating the right atmosphere for the drug distribution as observed for the buffered aspirin tablets. So however, certain buffer system it containing the potassium cation it inhibit the drug absorption as seen in the vitamin B2 and the sulfonamide. So colorant basically even we talk about the low concentration of the water soluble dye that can have a inhibitory effect of the dissolution rate. So dye molecule it get absorbed into the crystal surface of the drug and it inhibit the drug dissolution. So more the colorant has been used, more erratic the bioavailability is. For example, brilliant blue. So brilliant blue is basically hydrophilic dye. It retards the dissolution of sulfathiazole. Next category is the complexing agents, the complex formation that has also been used to alter the physicochemical and biopharmaceutical properties of a drug. Example including the enhanced dissolution through formation of a soluble complex. For example, the ergotamine tartrate. So ergotamine tartrate, the complex form with caffeine and the hydroquinone with the complex form with the digoxin. It improves the lipophilicity for better membrane permeability. Other example including the caffeine PABA complex, which improve the solubility and the bioavailability as compared to caffeine alone. So nature and the type of the doses of some apart from the selection of the doses regimen, selection of the dosage type. So the nature is also incorporating a better alternative to improve the bioavailability when we talk about the selection. As a general rule, the bioavailability of a drug from various dosage form like decrease in the following order. So solution exhibit higher solubility and the bioavailability, then emulsion, then suspension, then capsule, then tablets, then coated tablets, then entirely coated tablets, and then the sustained release product. The sustained release product it sustains the release. So the bioavailability and absorption is quite reduced as compared to the solution form. Product age and the storage condition. So the product aging and the storage condition can adversely affect the bioavailability by change in the physicochemical properties of the dosage form. For example, the when we talk, we when taken the product for a longer period of time, the precipitation of the drug in the solution was initiated. So precipitation, when we talk about the innovative precipitation, it leads to toxicity of the product and unchanged form of the drug basically excreted in the urine. So lesser amounts of drug available for the absorption and leads to poor bioavailability. Hardening of the tablet is also a major problem, and change in the particle size from micro to macro range because of the coalescence or the coagulation. So the questions that come in the topic, that is it basically what is the influence of the pharmaceutical excipient on the drug bioavailability? Give some examples of the complexation used to enhance the bioavailability of the drug. What is the disintegration time is not considered as a guarantee of drug bioavailability? What is the influence of the compression force on the drug dissolution and the absorption from the tablet, which is unpredictable? This is basically a tricky question. The example is the answer lies that the compression force, basically the upward compression force or downward compression force, it creates a mass transfer from the solid state to liquid state. When the mass transfer is initiated, the dissolution and absorption from the tablets is unpredictable because the binder and disintegrant interfere with the absorption. The compression force on the drug dissolution and absorption is always unpredictable in nature. So some give the some examples of the complexation used to enhance the bioavailability. So what are the nature and the type of the dosage form it influences the drug absorption, and what is the list of the orally administered dosage form in order to prevent the bioavailability problems. So these are some references. I have taken the literature data from Bermanker and the Sharjal book. So thank you very much for your kind attention. So any suggestion you can contact with me at a listed address. Thank you very much.